Welcome, everyone. Thank you for joining our Human Islet Research Network, or HEARN, webinar series. Today's presentation is going to be about FAIR data and you. Are you ready for 2023? I'm Joyce Nyland, co-PI of the HEARN Coordinating Center, which we call HIREC, the Human Islet Resource Enhancement Center. Yeah. John Cadiz is my co-PI. And we're both going to be giving a bit of an introduction today.
After an hour.
If you could all mute if you're not speaking. And our presenter today is Dr. Jeff Grethe from UC San Diego. And he's an expert in this area, and he's going to help you learn how to succeed with your next NIH grants. He's going to tell you all about the new data mandates of NIH and the opportunities that you have for enhancing your data and data sharing. So let's go to the next slide, please. Just want to give you a bit of a history and introduction to the HEARN. For those of you who are not fully familiar, it began in 2014, and it's an expanding, morphing network of consortia that now consists of 5 different consortia plus the HIREC, which is our coordinating center that we run at City of Hope. We have the Consortium on Beta Cell Death and Survival, or CBDS. Then there's CMI, the Consortium on Modeling Autoimmune Interactions. CHIB is the Consortium on Human Islet Biomimetics. We have a Consortium on Targeting and Regeneration, CTAR, and HPAC, which is the Human Pancreas Analysis Consortium that does a deep dive of phenotyping on pancreata. In total, now there's over 200 investigators involved, about 85 institutions, and it spans over 9 countries. So it's really growing and expanding. And I'll tell you a little bit more about the getting involved with HEARN in the future. Now, there are many benefits of a network such as HEARN. The goal is to accelerate progress in type 1 diabetes research. And this consortium structure gives a lot of benefits, such as early sharing of discoveries, attracting new talent. We have funding opportunities for junior and new investigators. We can develop new technologies and test new ideas, fostering collaborations through networking. We help to train the next generation through HEARN, and then there are interactions all across the diabetes and islet biology community via this consortium structure. So it really is a way to reach out across the diabetes research community, including things like this webinar today. If you want to learn more about HEARN, we just published recently the editorial that begins a series that will be appearing in Molecular Metabolism. Here's the first article we authored with our TNC, the Trans Network Committee, about the HEARN. It's just a few-page introduction from which that figure was taken. So you can look that up there. There's already the first in the series on what is an islet cell has been published, and several others, about another 10 articles will follow soon over the coming year. Next slide. And as I mentioned, if you want to get involved with HEARN, keep an eye on our website. This is the URL here at the top, hearnnetwork.org with just one N. Funding opportunities will be posted here for NIDDK and other NIH diabetes-related initiatives. And if any new funding opportunities for HEARN directly come up, we will be posting them here so you can learn more about applying. And that's how you could become a member of the HEARN, is a successful application. Next slide, please. And now I'm going to turn it over to my co-PI, John Cadiz, who's going to tell you about some of the additional resources that we offer through the HEARN. John?
Great, thank you, Joyce. So one of the bubbles that Joyce showed you was one that read early sharing, and this is one of the ways that HEARN is doing that. This is a tool that you can get to from the website, where bioreagents, data sets, documents, technologies are shared with the research community. So how is this sharing done exactly? There are 18 different categories of resources listed on that browser site. Here I'm highlighting for you 6 of the major ones. And what we've been doing during these monthly webinars is selecting one or more of these resources and taking a closer look at each, understanding how they were made or used and how it might be applied to your research. Now, in case you've missed those, these webinars are recorded and you can go to the HEARN YouTube channel to watch any one of those webinars. Now, the subject of today's webinar is more about how to share, the process of sharing, and what's changing. So I briefly wanted to address resource sharing in HEARN because it does take some effort, as you can imagine, to put these resources up there. Now, when NIH designed this network back in 2014, in each of the RFAs, there was this statement that was included. And this statement up front was one in which all of the applicants understood that resource sharing was a priority. And moreover, even within one of these consortiums, there was an additional program that was created, and it was exclusively a resource generation program. So essentially, its job, HPAP, which was part of the human pancreas, which is now part of the Human Pancreas Analysis Consortium, that program was created to exclusively focus on making these data sets and images available to the rest of the research community. So today what we're going to do is we're going to step through a series of questions and considerations to understand what's changing from NIH's perspective in terms of data sharing and what we can expect here in the future, understanding our research output, because there may be some things that you may not think of as a research output that is important. We're going to learn about these FAIR principles. You may or may not have heard that term FAIR. It means findable, accessible, interoperable, and reusable. And we're going to learn more about those today as well. We're going to look at some data repositories that might be relevant to your research. And then finally, we're going to consider the importance of data management and how that applies to FAIR. And then at the end, we're going to have a discussion. So while I stop sharing my screen and ask Jeff Grethe to pull up his screen, I'm just going to give a brief introduction to Dr. Grethe. He will be today our speaker today. He's a mathematician and bioinformaticist with over 25 years of experience in biological sciences and neuroscience in particular. He leads a FAIR data informatics lab at UCSD, where his group is involved in at least 6 large high-profile data-intensive networks. Jeff, you might be telling us more today. Dr. Grethe actually co-authored the FAIR data principles in 2016, and that paper has been cited 5,500 times at least. So that highlights really the importance of these principles in the scientific community. Jeff, so it's a real pleasure to have you speak to us today. I've learned a lot from you over the years and look forward to your webinar.
Thank you very much, John. Everyone can see my slides?
Yes.
Okay. John basically already covered sort of the topics for today. So what I'm going to do is walk you through sort of the new mandates and the sort of the FAIR principles and how they apply. But I wanted to first give a little bit of information about the project that is driving some of our work in the NIDDK space in terms of providing guidance around these FAIR principles, and that's the NIDDK Information Network, also known as DKNet. And really, we're a project funded by NIDDK to be a research resource information portal for biomedical researchers, and to help connect DK researchers to various resources and centers, and you'll see down in the presentation, there are a number of tools that I'll mention from DKNet that can point you to resources around data sharing and the data mandate. On the website itself, if you're looking for information on the data, there's a little breakout box in the top portion where it talks about fair data, and so some of the content I'll be talking about today is available there, we also will point to the HEARN webinar, through our webinar channel. So, when people talk about data sharing, a lot of times the concept of open science is brought up, and really this is about, increasing access to, as John mentioned, the data, the protocols, the reagents, to make sure that, those are available, but that also that the information is transparent in terms of how it's described, so that, for example, even when one looks at protocols, a lot of times in the literature will say, this protocol is adapted from some other paper. You go trace that paper and you trace 6 papers back and you still can't find a description of the protocol. So making sure that things are transparently described and as open as possible, and to make it easier to communicate the scientific knowledge. And this idea of open science and data sharing, has been something over the pandemic that has shown to really accelerate research. There are a couple of papers here on this slide that sort of describe how this has, so the pandemic has sort of taken hold of open science and data sharing to really push research in the space. And so I think that's sort of an interesting outcome, of the pandemic in terms of supporting some of these principles. Motivations, for open science and a key part of that again is data sharing, is that we are still sort of ramping up a digital revolution where it's not just access to data but access to computation as well. There's additional computational capacity that can be used to mine biomedical literature and data for new insights. And so making more data available to these machine learning or AI techniques, as a lot of times they're mentioned, is critical, I think, for the future of science. And then the other areas that research is largely funded by the public, and the public should have access to these results, and should be made available to other researchers. There's also trends in open data. So a lot of people know about sort of these big science projects that generate large pools of available data. So this is the original human genome, but now there are many projects looking at various aspects of data. So the Spark project within NIH that I'm a part of is looking at anatomical connectivity in the peripheral autonomic nervous system and information about the end organs, kidney, liver, and such, and providing data. It's a data generation project first, making sure that data becomes available. But there's also this long tail of data where there are claims made in papers, and the data that supports that can also be of use in generating new insight, and it can also be used to aggregate some of these long tail data to increase analytic power, and to do analyses that weren't available to be done before. And lastly, I think the trends and the title of the webinar leads to funders and journals are increasingly requiring data sharing. So this is something that is also becoming more of a requirement, and will continue to over the coming years. And so that brings me to the new, the final NIH policy for data management and sharing. So this is the update that was published earlier this year. So this was a multi-year process. And the high-level takeaways is that the new proposals will have to have a data management and sharing plan. So this is description of data management and sharing, not just the resource sharing plan, but you also have to describe the data management during the life of the proposal. All data must be managed in your lab, but most data should be shared. And a key principle is as open as possible and as closed as necessary. So, this takes into account, issues with, protected health information and sensitive data. So some of the details for the policy. So for any competing grant applications that are submitted to NIH after January 25th, 2023, and any subsequent receipt dates, need to follow, this new policy. And then there are some other dates, so contracts, and intramural research projects, are also the January 25th date, but there's slight differences. So, for example, the intramural research projects conducted, after on or after January 25th, fall into under this policy as well. So in terms of the details, the what, so, what constitutes scientific data? And so this is something that was actually discussed fairly deeply, during the formulation of the policy. And so basically, the scientific data is going to be the factual material, that can be used to validate and replicate the research findings. So that's, and for example, imaging data from spreadsheets, so anything that can be used to validate and replicate the research findings in a publication. What they clarified is that the scientific data does not include laboratory notebook, completed case report forms, drafts of scientific papers. So, since data is a very open term, they made sure to clarify that this mainly pertains to the data that was used to generate the research findings. So the primary data that was used to generate the research findings, and not associated information such that might be kept, for example, in a laboratory notebook. The when is another important part. So according to the policy, shared scientific data should be made accessible as soon as possible, and no later than the time of an associated publication or the end of the award support period. So if you are generating data and you publish on that data, it is required through the policy that that data is made available at the time of that publication. And many repositories support sort of embargo periods where you can work on bringing your data to a repository while you're also working in terms of getting the data set or the manuscript published, so that they can appear at the same time. And also that what I'll talk about, identifiers, the digital object identifiers, can point to one another. The where, which I just mentioned, is that the policy encourages the use of established repositories to the extent possible. And I'll talk about repositories as well further down in the talk. In terms of what is specified, NIH encourages practices that are consistent with FAIR. Another change, which is important, is that the funding component of this is that fees for long-term data preservation and sharing are allowable, but you must pay for those fees during the performance period of the award, even if those funds are to be used, for a longer term period of data storage. So this is something different from some normal NIH awards where you can't pay for services past, the date of the current fiscal year you're in. Here, NIH has pointed out that you can pay for services beyond the period of the award. specifically for data preservation, but those funds must be spent during the performance period of the award. And then there are also some, if one doesn't follow one's data management and sharing plan and data is not being made available, NIH does have some ability to, will have ability to follow up in terms of a non-compliance. And, in the policy, they talk about, NIH for future funding decisions for the recipient funding institute, for the recipient institution. So again, those are sort of the highlights. There are, a number of aspects of data management, that are important, to make sure that data that you are making publicly available, so that it isn't that much of a burden at the end of the project period or when you do have a manuscript that is published. And so this is something, that, when you're starting to plan your project, having, for example, detailed plans about, how you will manage data, and that you might actually need to revisit over the course of the project. For example, if the data that's being collected, some of that changes or the process changes, these sort of plans can also help in terms of optimizing future data collection in the lab. So basically having well-detailed data management plans for the lab will help you with being able to organize the data for future sharing, but also help your lab in terms of optimizing that for collection throughout the studies that you're doing, and then also having a way to organize that over time in the lab. And data management also need to have policies for saving and backing up your data. So this is another critical aspect. A lot of institutions do have support for data backup. There are many different ways that can be done, but that is something critical. And there is actually a good paper that describes sort of supporting your data. So Research Data Management Guide for Researchers that came out in 2018. So I provide the link here, but that provides some good core principles for data management. And good data management actually impacts a lot of different constituents. So in terms of science and data sharing, that's sort of one thing that it helps with, and that's sort of I guess the broadest goal. But there are a lot of ways it helps both yourself and your future self, within and within your lab itself. So if one has underpowered studies, being able to aggregate information from prior studies, and this has been shown in, for example, imaging studies where one can bring back data, that was collected before, and make that useful in the context of a current study. Without good data management locally, finding and using prior data, especially if it's not well described, becomes very difficult. In terms of the future self, one thing my colleague Marianne Martone has mentioned is that she has never regretted annotating her data too much. She's only regretted it when she hasn't annotated it enough. So not being able to understand exactly what data you have, and also the lab benefiting from data that was collected in the past, either for additional student projects and the like. So being able to access that past data can be good. And then also the sharing with colleagues, and being able to document the data as graduate students and postdocs cycle through the lab. There may be a time when one needs to be able to look at the data to revisit potentially some analyses or use data in a subsequent analysis. So how do you do that? There are different ways. Again, data management is the key part of that. But in terms of the sharing of the data, is the using of the established repositories for sharing of data. And this is where sort of the FAIR principles come in, as these repositories make their repositories more fair and to support fair data. And as John had pointed out, the high-level principles are really to make data findable, accessible, interoperable, and reusable. The principles themselves are very high level, and sort of the implementation of these principles is left to the communities and the data repositories, as some of the issues with metadata, for example, are really going to be defined by the communities. But there are some high level concepts that sort of cross all communities, and that actually applies to the idea of FAIR, is that it's not just for humans. Since we are dealing now with more computational tools, more access to data via computational tools, any of the metadata, any of the information that's being provided for data sets, should be able to be processed by a computer without human intervention. And so this is really where it gets to the concept of machine readable data. And this is basically human readable data that is marked up so it can be read by machines. And so there are various microformats and standards around that. And so that is something important to keep in mind, especially, for example, this is just a simple sort of example, but one of these is actually a data file and the other is an image of a data file. So the data file itself, the CSV file, the Excel file, can actually be used by a machine, can be operated on by a machine. The image, not really, unless you have to put a lot of machinery, to basically extract information from a table, and that is not 100%. Conversions like that are not guaranteed to be 100% reliable, and so that's why having the data as an actual spreadsheet is machine readable is important. Now, a key thing about FAIR and the description of data is the metadata. And so this is really, the data about the data itself. And sometimes, this is sort of there's no hard boundary. It's sort of a gray area in terms of, what some people may consider data, maybe someone else's metadata. However, making sure that your data sets are described to a sufficient detail, for future use is key for any of these data sharing mandates. And I'll go through some of the different qualities for metadata, and take a closer look at FAIR. So FAIR again is a very high level principle, and it's sort of like apple pie, mom and apple pie, in that it's all sounds well and good, but we need to make sure that the details are implemented and follow to basically enable what FAIR promises. And under each of the sort of FAIR attributes, there are a number of sub bullets that describe specific aspects of FAIR that are important. And I'll quickly walk through some of them. So under findable, this is really about being able to gain access to the data. And so the two clear primary aspects of this are that data are assigned a globally unique and persistent identifier, and that data are described with rich metadata. So what is a globally unique and persistent identifier? And this is possibly the one of the most important aspects for FAIR data. is that there is a unique handle in the world that is able to get someone to that data set. And so that is important, since this data set, is going to be described potentially in journal articles and preprints, will be referred to by other data sets. And so there needs to be a stable, persistent identifier that points to that data set that was used. And so it's identifying that single data set, that single object, so it isn't reused. And it needs to be persistent so that, 10 years from now, if your local website goes down and that website URL, is no longer there, that we still know what that data set is about. And this brings into play important aspects around ability to see the metadata for a file, even when, for example, a data set, 30 years from now may not be located, we can still get access to the metadata to understand what the data set was about. And so there are a number of persistent identifiers. So everyone should probably knows the digital object identifier that one gets, when you have an article published in the literature. There's the ORCID identifier, which is identifiers for researchers. There's a research identifier, which a lot of journals are now including to identify resources used in studies. And these are globally unique. They're all persistent, globally unique identifiers, and are issued by various registries who track and identify these unique entities. And so for data sets, really the identifier that is, the most common in terms of, providing this persistence is also DOIs. So it, this is an example, from, the HEARN resource browser for a data set, that has been shared, and it has available DOI. This is issued by authorities such as DataCite, and links into Crossref, which manages, digital object identifiers for the journals. And these are also issued by data repositories. In addition to DOIs, databases also have accession numbers, which are stable and unique. So for example, GEO, issues, its identifiers for its data sets. And so that's another type of identifier that one might run into. And, the management of these identifiers is a social contract. The idea is that, you will never get a 404 not found when one looks for a DOI. You will always get the metadata related to that data set or that journal article. And then, if the data is available, in the repository, it will link back to that data as well. There are, benefits to using a trusted repository because they do provide these persistent identifiers. And so you shouldn't be assigning these to your data sets. And so again, looking for suggested data repositories, through DKNet, we do have a guideline, some guidance for data repositories, and that is something that you can visit DKNet for. But again, this is something you should not be doing yourself, but you should be looking at repositories for that. The next part was describing, the data sets with rich metadata, the concept of rich metadata will change over time. As new standards, new information, is gleaned as to what is important for data sets. That will continually evolve, over time. However, we do know, when we publish papers, that there are, some very standard, aspects, of a research article, and that is something that, is being defined actually for data sets themselves. So, PIDs plus rich metadata, is really creating fair data sets. So this is an example from are the Spark project, which I mentioned earlier, going through, some of the data descriptions and things that are included, as part of the metadata, for this data sets. So there's, for example, a full experimental protocol defined at protocols.io, that provides a full protocol, for the data generation. There's a data description, so an abstract, but really about the describing the data. So that's the study purpose, but then data collection, and a hint at what was concluded in the research article. Links back to the originating publication. And links to any code that we use to analyze this information. So again, this rich metadata is being defined in many communities for these data sets. And a lot of repositories are implementing various rich metadata schemes for the data. And so again, more metadata is always better than less metadata. And really, what needs to be kept in mind is what information do people need to understand the data and use it. So that is something that's important. So the A in FAIR talks about accessibility. And here, accessibility talks about using standard protocols to retrieve data. Again, repositories will handle that for you. But there's also the idea that the access to the data may need authentication and authorization. And again, this gets to the topic, as open as possible, as closed as necessary. So when people talk about fair data sharing, it doesn't mean that all data is going to be, freely open to anyone. There are going to be cases where data needs to be protected, and we'll have to go through certain authentication and authorization, for access to that data. Again, this, we talk about, the access to the structured versus the unstructured metadata. So again, in a lot of these repositories, they require structured metadata to be entered with the data set. And then this can be easily packaged up, and made available, through, various machine readable aspects. And so, as an example, you may have unstructured information, subjects comprised, male and female, and, the description of the organism. But, it's really, when one turns that into structured metadata, it makes it much easier to be searchable through a repository, and allows, again, enhanced usability of the data, through the repository, where it might be found. The interoperability aspect of FAIR talks about the use of sort of accessible and broadly applicable languages, for knowledge representation. And so talking about standards that are used, and also the use of vocabularies, controlled terminologies to describe information about a data set. So for example, in a research scenario, you might have, different laboratories studying, a rat model of type 1 diabetes. So you have 3 different data sets. You start looking at the metadata for this data set, and now all of a sudden, things are less clear, right? So for example, if one looks at location, there are, 3 different descriptions, and the last one that says cortex, that is very broad, very generic. And what does that actually mean in this context? Even though, it is, detailed metadata, the actual content of that is may not be as understandable as one might first think when one is collecting this data, because a lot of time data collection in the lab is very context specific. So everyone in the lab might know what cortex means in regards to this data set, but someone outside the lab may not. And so the use of FAIR vocabularies, which also have these persistent identifiers and have additional metadata, become important. So for example, describing the data set as cortex of the kidney, has a specific terminology identifier that can be both used by humans and machines. And a lot of repositories will handle, generating some of these mappings when you add metadata to them, or something that would need to be implemented at the lab. And these identifiers are important to unambiguously identify these entities across multiple data sets. And so it applies to reagents and tools. So which antibody was used. It's a lot of times the textual descriptions that we give to reagents are not good enough to uniquely identify them and to get back to that specific antibody, especially if catalog numbers change, and antibodies are sold from vendor to vendor. The use of gene IDs, there are many different identification schemes there. So IDs come in play in a lot of parts. And so, for example, in describing the organism itself, there are the research resource identifiers that one can leverage. In terms of the gene identifiers, in terms of describing, what is the information about these data sets, even though the textual information and the textual notations may be different, they point to the same identity. And then clarifying, again, in this example, the anatomical structure where you have these anatomical identifiers that specify what was specifically targeted, where the first two are pointing to the same anatomical entity, and the third one is actually a completely different entity that is being described. And so that's where these persistent identifiers come into play in terms of disambiguating and making clear, what research data is about. And so, as an example, the research resource identifier is really used to target resources that are used in a study. It is something that we provide resource reports on within DKNet, so we're able to provide information about these resources. And because of the use of persistent identifiers, we're able to tie together a lot of information around these resources. So provide, information on validation studies or ratings, who has used a specific research resource before, alerts, for example, problematic cell lines, that one might encounter, having a system that uses these persistent identifiers allows, systems like DKNet and others to pull and aggregate this information together and make that available to researchers. And so basically, when you're collecting information, make sure, that, when you're starting up a study, and getting information about the reagents used, the model organisms, any of the data sets, that, if it's a study that you're also looking at other data, having the identifiers for those data sets, it's important to manage and include all that information. And the last aspect of FAIR, really talks about reusable. And so this is, that the metadata, richly describes and has, plurality of accurate and relevant attributes, and also that it meets community standards. And so one of the core points here is that when you start managing your data and managing the information about a study, it's important to think about where the data will end up from the beginning. Because there are a number of repositories, and repositories, domain-specific repositories for data, like GEO at NIH and other repositories available through the community, have specific metadata requirements. And so, as I mentioned, you can go to DKNet and find information about data repositories that are available, and we provide some information, for example, about what it takes to download or upload data, and also link to information at fairsharing.org that provides information about the data requirements. And each data repository is different, right? So we've been involved in a study where we basically looked at databases to see how compliant they are with the FAIR principles. And again, this is something that is evolving over time as repositories work towards becoming more fair. And, one of those aspects is, implementation of standards at repositories. I mentioned fairsharing.org. And so this provides the listing of databases and the standards that are implemented by those repositories. And again, this is important metadata, that's included in these standards that, you will need to make available when your data is shared. So, this aspect of data management, making sure that you look into the future a little bit, and that's why the data management and sharing plan is important. As you outline, what repositories you will be sharing your data at. And again, it could be multiple repositories. So for example, if you're collecting imaging data and, some, array expression data, you may be, depositing to an image archive and also geo. So you need to keep in mind that, it may not just be one repository. You may be required to submit to multiple repositories and to make sure that you're collecting the appropriate metadata so that, 3 years, 4 years down the line, you don't have to scramble and try to figure out, what was, this bit of information that you needed. And there are, many tools to help with implementing, sort of your data management and sharing plan. So one of the ones, the MIT libraries and, their online courses that provide information. There's also a tool, the DMP tool, which provides you with the ability to use templates and to be guided through the generation of the data management and sharing plan. And again, the content in the DMP tool is being updated for the 2023 deadline. So that's another very good resource, for people to know about. The other thing that I want to touch upon is, the provenance for a data set. That is that, as I pointed out before, where you link out to a specific experimental protocol or tying into associated code, there's also, tying into information about the originating publications. And then also when people cite data, that will also be an aspect of this provenance. So, the use of a persistent identifier like a DOI is actually tracked in systems that are implemented now for citation tracking. So Crossref, does this for DOIs. And so this is something, as, data sharing and publishing matures, that these systems will be picking up these persistent identifiers, and that really gets to treating data as a research product like a publication. And so there are a set of data citation principles. So this is Joint Declaration of Data Cite Principles that were put together by Force 11 and have been endorsed by many journals and organizations. Really provide information as to how data should be cited and how that is formatted. And these data citations, so this is an example from one of the repositories that we run, where the data set citation is very similar to a regular citation, including the DOI, but also includes for example, the local identifier in the repository, but is something that would, looks very similar to citations that people are using now. And as I said, these are now being picked up. So, for example, in Google Scholar, it will actually say how many times that data set was cited, and we'll tie that into Google Scholar. So again, by using these persistent identifiers, DOIs, using the repositories that provide those, you're really tying into a whole ecosystem, that brings data, into sort of the same pipelines that we're tracking journal articles. So for a lot of this, it is overwhelming, but one needs to remember that this is a partnership. So it's, there's three sort of key partners that are dealing with the data. So the researchers, the repositories, the index aggregators, and then there are other projects such as HEARN and DKNet that try to help guide researchers and help provide, tools and best practices, within communities to meet these mandates. And so again, repositories and registries, really help, with providing the persistent identifiers, providing the machine readable metadata, ensuring the data is in a good format. The index aggregators such as Google and others, provide for search across that, and they rely on the repositories and registries to provide this machine readable metadata. And then the researchers, need to, prepare for data sharing through, good data management, the preparation of rich metadata in terms of preparing to share all this, and using the appropriate community standards used by the repositories and by the scientific communities in terms of structuring that data when it is shared. And so again, if you have any questions, you can always come to DKNet. We have material online. You can always ask us, send us email as well. And we're also in the process of developing additional help with fair data. So there is going to be a data wizard, to help guide people through some of this to make it a little easier to pinpoint repositories that may be of use. And so again, we are all sort of changing the culture of biomedicine data sharing one step at a time. We have begun, but we are not, we are not finished. And again, this is something that is going to evolve, over the coming years, and especially, in terms of the repositories that are supported, by NIH, either internal or external to NIH, and also, by other funding agencies. So, for example, Elixir in Europe funds a whole number of repositories as well. And so this is something that, that landscape of repositories is going to change over time. But it is something, that is, there's a very rich ecosystem now, and it's only going to get richer. So I would like to thank you for listening. And again, I guess we now turn into the discussion section. So John, I'll turn it back over to you.
Thank you very much, Jeff. That was a terrific talk. There's lots of detail there for us to take in. Let me pick up the first question that is coming from Brian Main. Jeff, I also found an interesting point that you said that NIH would now allow us to spend in our grant period for the cost of storing data for some period of time. And the question that Brian is asking is, can you speak to the ongoing cost of storing data? Because, let's say I pay for something for, 10 years, but what happens after that 10 years? Who knows? But I think maybe Brian's question is just more about the general cost of storing data. Can you speak to that, please?
Yeah, I mean, I think sort of the technological side is that storage costs are decreasing over time. I mean, that's sort of a given, and certain repositories are using different models. So there's some repositories where, for example, if you're funded by NIH for certain work, for some imaging work, that repository, is meant for that data and will actually host the data without the need for payment. But that, again, this is evolving and will change over time because NIH is always talking about sustainability models for these data repositories. There are other models that are out there. So one is difference between small and large data. So there are some repositories where if it's under a certain size, so a couple gigabytes, then it will sort of be free to make that data open. But for larger data sets, they charge by the data size, but it's a one-time cost, right? So again, this gets to the data will be stored in the repository for X number of years, depending on what the repository's terms of use are in terms of these data sets. But you're paying an upfront cost for the data set itself. There's also another model and this is actually implemented, I think, by Dryad with some journals, is that when a data set or when a paper is published with an associated data set, that there are some publication fees that sort of that get shared between the publisher and the and the repository itself. And I'm not quite sure exactly how that's all handled, but again, everyone's trying to figure out the sustainability model for repositories going forward. The most common one with charging was the first one I talked about, where you sort of have an upfront cost, and that's then for a number of years for that data set, potentially in perpetuity. So I think it was a university library that had that policy where they charge up front, but it's sort of the data will be held in perpetuity by the university library. But again, it's an upfront cost.
Yeah, thank you, Jeff. The second question I think I can answer. This is coming from Sasenka, who found your presentation very helpful and wants to share it with colleagues that may have missed it. Sasenka, we are going to post this on our YouTube channel, so you can go there and you can review the presentation at your own leisure. The third question
you can also have if you want, you can also have the link to the slides if people want to, download the slides.
Great. Thank you, Jeff. So we'll go ahead and enable that as well. The third question we have is coming from Marcella Brisova at Vanderbilt. And Jeff, she's a big imaging person. So she's curious about NIH's plans for imaging data repository analogous to the IDR in Europe. Do you know anything about that? And can you comment on that?
Yeah, I mean, there are a number of data repositories that are supported by NIH right now. So there's the through the BRAIN initiative, there's the BRAIN Image Library, which is at the Pittsburgh Supercomputing Center, I believe. There are other resources such as the Cell Imaging Library, which also is a place for the publication of imaging data. And so there are a number of repositories in the US, sometimes focused more on certain communities around imaging data. I'm not that familiar with IDR, so but hopefully that helps in terms of your question. So if there's anything more specific, you can always contact me via email as well.
Thank you, Jeff. Jeff, I wanted to ask a question. I recognize many names on this call, and I know that there are many people on this call that have their own databases, have their own large data sets. One thing I found interesting was this paper that you have, I guess this preprint server where you've evaluated essentially fair compliance to these guidelines. Yep. Is there a generalist sort of tool that people can use to run their databases or systems through this, or does one have to kind of go through each of the components that you shared with us individually to come up with some kind of an assessment?
Yeah, so we do have an assessment tool that we're using internally. I mean, if there's any repository that wishes to run this on their own repository, we'd be more than happy to provide that information to you on how you can access that, and sort of review your own repository. But yeah, I mean, again, the implementation of FAIR at repositories is, as I said, is evolving, and it is going to evolve, as communities define sort of what FAIR means to them. Because again, they're guiding principles, but the actual details, are left to the reader.
Great. Well, thank you very much, Jeff. I really appreciate it. We're nearing the top of the hour. So again, I want to extend my thanks for preparing this presentation and sharing with us a lot of good information that we can use and will certainly value in the future as things change.
Yes, thank you very much, Jeff. We appreciate it.
You're welcome.